Assalamualaikum dan salam sejahtera. Dalam video ini, saya akan terangkan tentang chapter atau topik 9, Reproduction dan Development. Reproduction and Development. So, in this video, I'm going to explain about 9.1, Sexual Reproduction in Flowering Plant. The learning outcome from this subtopic is for students to be able to state and define the terminologies involved in gamete formation in flowering plants. Then, students should be able to explain the development of a pollen grain and formation of male gamete. Then, students should be able to explain the development of ovule, embryo sac, and formation of female gamete. And then lastly is for student to be able to explain double fertilization in the formation of seed. Let's look at the first terminology. It's microsporangium. From the word sporangium, angium means a sac, a structure that contains diploid microspore mother cell that will later on will develop into pollen grains. So the structure that contains diploid microspore mother cell is called microsporangium. So let's look at the terms micro. In reproduction, micro will always related to male. Related to male. So this flower has, this part is the anther. So if we cut the anther, we will find the microsporangium. So this is the microsporangium. This whole structure is called anther, and inside the microsporangium is the microspore mother cell. So this is pollen sac, pollen sac, which is also known as microsporangium. The pollen sac will develop into pollen grains. Okay, the microsporangium. The structure that develop into pollen grain is the microspore mother cell. So microsporangium will contain a lot of pollen grain later on. So inside the pollen grain, there will be two types of cells, which will be explained later. The second terminology is microsporocyte. Microsporocyte is the cell, right? You can use the term microsporocyte or microspore mother cell. Both are the same thing. So microsporocyte, which is diploid, can be found in the microsporangium. So this microsporocyte, the microspore mother cell, will undergo meiosis. So as we all know from cell division topics, meiosis will produce 4 daughter cells. So the daughter cells that arise from meiotic division of microsporocyte is called microspores. Microsporocytes, let me repeat. Microsporocyte is the diploid cell in the microsporangium. It will undergo meiosis to form 4 haploid microspores, or also known as tetrad. Tetrad means from the word tetra. It means 4 cells. During cell division, we learned that tetrad is referring to the 4 chromatids. Okay, so you find this word again, but it is referring to the 4 haploid microspores. So this microsporocyte in the microsporangium undergo meiosis and produce 4 microspores. So these are all microspore. The microspore is the next term. Microspore will develop into pollen grains by mitosis. The third terminology is microspore. After mitosis, the microspore will develop because mitosis produce two daughter cells, isn't it? So after mitosis, the microspore that undergo mitosis will produce two types of cells. One cell is the generative cell. The second cell is the tube cell. So this is already a pollen grain, but it is not a mature pollen grain. See? This whole structure is called pollen grain. Inside the pollen grain inside of the pollen grain, we can find generative cell and tube cell. So the fourth terminology is pollen grain itself. So what is pollen grain? A pollen grain is a microscopic structure that contains generative cell and tube cell enclosed within thick resistance wall. So the wall must be um, uh, must be able to withhold any pressure for the time being, because they need to be transferred or pollinate another flowers. They need to be able to be light enough but able to protect their cells in the pollen grain. So usually we can find that from observation, pollen grain is usually have an outer layer that is spiky. Dia ada tajam-tajam. Kenapa? Sebab debunga ni, pollen grain is called debunga in bahasa Melayu. So debunga ni perlu melekat dengan pollinator ataupun agen pendebungaan. Jadi nak melekat dengan agen pendebungaan untuk dia kemudian tersebar ke bunga lain ataupun ke female structure of another flower. Jadi bila dia melekat dan kemudian dia akan jatuh di atas stigma. Jadi for them to be able to stuck on their pollinator, they need to have a structure that help them stuck on the pollinator. Jadi nampak biasa bunga ni, kalau awak tengok kat bunga raya, adalah parts yang ada warna kuning-kuning dan dia memang sangat halus. Dan kalau kita observe, kita buat pemerhatian di bawah mikroskop, kita akan nampak struktur yang begini lebih kurang. So this is the outer layer, which is the thick resistance wall. Dia mampu menahan macam air, lembapan air yang terlalu banyak, so dia boleh tersebar oleh angin dan juga air. Mungkin lalu bila ada air hujan mengalir, dia boleh bergerak boleh mengalir dari satu bunga ke satu bunga yang lain. The fifth terminology is generative cell. Generative cell will divide mitotically to form non-flagellated male gametes. Someone asked me about what does it mean by non-flagellated, and another word is non-motile. So when the sperm, the male gamete is non-flagellated, meaning they cannot move by themselves. They cannot swim. So they need some structure to help them arrive at the female reproductive organs. So male gamete of flowering plants do not have flagella, so they are non-motile. Non-motile is unable to move freely. The generative cell will divide. Of course, mitosis will form two types of cell. So these two cells, both cells are sperm cells. So in flowering plant, there will be two sperm cells, while the tube cell will form pollen tube. So pollen tube is a very important structure that helps sperm to travel to reach the ovule. Ovule is the female reproductive part. Okay, now finish about male reproductive organ. We get into the female gamete formation or female reproductive organs in flowering plants. The first term is Megasporangium. Sporangium again, it's a structure that is holding a female reproductive parts. So it contains a megaspore mother cell or megasporocyte, also known as megasporocyte. So from this flower, this is the sepal. The petal. This is the sepal. I think this is the sepal, the petal, and this is the dia terbalik bunga ni dia upside down. So this is the structure where we call ovary. Inside the ovary is the ovule. Ni bunga ni adalah bisexual because it has both male and female reproductive parts. Inside the ovule is the megaspore mother cells. Jadi ni yang belum develop into a very defined or detailed female gamete. Jadi ini adalah sel asal. The mother, sebab tu dipanggil mother cell. Megaspore mother cell or megasporocyte. Each ovule within an ovary of a seed plant contains a megaspore mother cell. Jadi dalam satu ovary ada more than one ovule usually. Selalunya ada lebih daripada satu. Ada juga tumbuhan yang memang akan ada satu saja ovule. So the megaspore mother cell is the megasporocyte. It's a diploid cell that will later on divide by meiosis to produce 4 haploid megaspores. Jadi ingat, meiosis will always produce 4 daughter cells. But in female, there is the difference between male and female. The development of the daughter cells after meiosis is in female. Out of 4 daughter cells, only 1 cell will develop into female gamete. So megaspore mother cell undergo meiosis and produce 4 megaspores. So only 1 megaspores will survive and undergo mitosis. So the third terminology for female gamete formation is megaspore. Megaspore will divide mitotically to produce embryo sac. So the surviving megaspore will undergo mitosis and it should produce 8 nuclei. So how many mitotic division should happen to produce 8 cells? So mitosis should produce 2 cells, isn't it? So each mitotic division should produce 2 daughter cells. To produce 8 nuclei or daughter cells, mitosis should happen thrice, 3 times. Okay, 1, 2, kemudian 1, 2, 1, 2, kemudian 1, 2, 1, 2, 1, 2, 1, 2. So 3 kali untuk menghasilkan 8 nuclei. Jadi in another word, megaspore will divide mitotically 3 times to produce embryo sac. Okay. Inside the embryo sac, we will find the 8 nuclei that I had been talking about just now. The embryo sac is a multicellular structure that has 7 cells with 8 haploid nuclei. There should be 3 antipodal cells and then a polar nuclei. This is 3, 1. And then 2 synergid cells and 1 egg cells. So 3 plus 1 plus 2 plus 1. It should be 7 cells. But then it has 8 nuclei because a polar nuclei consists of 2 nuclei. So the cell is a polar nuclei, but it has 2 nuclei. Antipodal cells has an unknown function. The antipodal is at one end of the embryo sac. And then in the middle of the embryo sac is the polar nuclei. Polar nuclei that has two nuclei. It is a large central cell of the embryo sac. And then the seventh term is the egg cell. Egg cell is the cells that will fertilize by the male gamete and form diploid zygote. So this is the one that will develop into a zygote, into a seed. Synergid cells is at the parts where it is close to the micropyle. Micropyle is the opening that leads sperm or the male gamete into the embryo sac. So synergid cells are two haploid cells that help to attract the pollen tube to the embryo sac. Sebab kita tahu katakanlah this is the stigma and the pollen grain landed on the stigma. So automatically when it landed on the stigma, it will be stimulated by the substance on the stigma. So it will be stimulated, pollen grain will be stimulated, and the tube cell will generate a pollen tube, while the generative cell will stimulated to divide mitotically and produce two sperm. So this pollen tube will find their way to the micropyle. So the structure that helps the pollen tube to lead the pollen tube to the micropyle is the synergid cells. So the overview of gamete formation is basically starts within the sporangium that contain the sporocyte, either megasporocyte or microsporocyte. So this microsporocyte or the spore mother cell will undergo meiosis and produce megaspore or microspore. Okay, ini secara umum kita panggil spores. So these spores will develop into gametophyte, and then this gametophyte will undergo mitosis. So in female, the megaspore will undergo mitosis, and then one of the megaspore will develop into gametophyte, undergoing mitosis 3 times. Under 3 times and it will produce gametes. Okay, 8 nuclei. While in male reproductive system, the microsporocyte will undergo meiosis and produce microspores. The microspore, all of the microspore will develop into gametophyte. Okay, undergo mitosis and produce pollen grain. And then pollen grain, it will produce two types of cell in the pollen grain, the generative cells and the tube cell. When it landed on the stigma, when the pollen grain landed on the stigma, it will undergo another mitosis and produce male gametes and pollen tube. The anther has 4 pollen sac usually, and memang struktur secara umum anther has 4 pollen sacs. In the pollen sac, we can find the microspore mother cells or microsporocyte. Each microsporocyte undergo meiosis, producing 4 haploid cells known as microspore. Each microspore will give rise to haploid gametophyte, male gametophyte, or immature pollen grain. The pollen grain will be mature when they landed on the stigma. So the microspore will divide by mitosis to produce generative cell and the tube cell. When the pollen grain landed on the stigma, it will undergo another mitosis to produce generative cell to produce sperm cells, while the pollen tube will develop into, the tube cell will develop into pollen tube. So the formation of sperm cells is when after the pollen grain landed on the stigma. Okay, it does not happen in the anther. It happens when the pollen grain landed on stigma. While ovule develop from couple tissues, which is the placenta that is held by a funicle. The young ovule is a cluster of parenchyma cells. The nucleus, this is the nucleus. Okay, in the ovule, in the ovary, in the ovule, there will be megaspore mother cells contained in the ovule. Now in the ovule. The Megasporangium in the ovule contain Megasporocyte. Okay, the megaspore mother cell. Each megasporocyte undergo meiosis to produce 4 haploid megaspores. Only one will survive, another 3 will degenerate. The one surviving megaspore will divide by mitosis 3 times, producing 8 haploid nuclei. Jadi dia kata kat sini without cytokinesis sebab dia masih dalam ovule yang sama tetapi hanya membahagi karyokinesis saja. Nukleus saja. Sebab tu kita panggil dalam tu adalah nuclei. Okay. Dan kemudian dia jadi sel walaupun sel sebab dia akan terbahagi secara fungsi yang berbeza. Two groups of 4 nuclei will move to both ends of the embryo sac. Okay, sebab dia masih dalam embryo sac. Kalau dia bahagi 8 dan cytokinesis, dia akan jadi sel embryo sac yang berbeza. Jadi apa yang berlaku ialah dia hanya menghasilkan nukleus yang berbeza. Okay. 4 akan bergerak ke ends north ataupun satu hujung, 4 lagi ke satu hujung. Each one of the nucleus will move to the center. So antipodal cell is left. There will be 3 antipodal cells left at one end, and then the synergid, one of the cluster at the micropyle will form female gamete, the egg. The polar nuclei is not partition. Kalau nampak kita tengok the 3 nuclei located near the micropyle will become egg cell, one of the 3 nuclei. 2 become synergid cells, and then the 3 nuclei on the opposite ends become antipodal cells. And at the center of the embryo sac, it will be polar nuclei. Eventually, one embryo sac consists of 8 nuclei contained within 7 cells. So this is considered as one cell with 2 nuclei. So this one, female gametophyte, ni mitosis, daripada ini develop into 4 haploid megaspores. Only one, kita kena tambah sini, only one megaspore develop into female gametophyte through mitosis. And then kat sini 3 times mitosis, and then last one it will produce female gamete, the egg. Kita tak panggil ovum dalam tumbuhan, untuk pembelajaran ni kita panggil egg cell. So what is it that you need to identify the name of the terms, and the second one is the ploidy level. So kita tahu ploidy level kat sini adalah diploid, and starting from microspores, it's already haploid. So selepas haploid mesti mitosis lah, tak boleh meiosis lagi. Flowering plants undergo double fertilization. So what is double fertilization? It is a process of fusion between one of the sperm cells. Ingat tadi, generative cell divide by mitosis to produce two sperm cells. So one of the sperm cell will fuse with an egg. It will form diploid zygote. The other sperm cell will fuse with two polar nuclei here. It will fuse with two polar nuclei. So two nuclei, each of which is haploid. When it fuse with a sperm which is also haploid, of course it will form triploid endosperm. It is called endosperm. It does not call a zygote. Dipanggil triploid endosperm. Struktur itu adalah endosperm. The triploid endosperm. While the zygote will develop into embryo. So for double fertilization, when pollen grains land on the stigma, and then it will be stimulated to germinate. The generative cell will divide into sperm cells, two sperm cells, while the tube cell will develop into pollen tube. Then pollen tube grows and leads the sperm cell towards embryo sac. When the pollen tube penetrates the wall of the embryo sac through micropyle, the sperm cell will enter the embryo sac. So one of the sperm will fertilize the egg cell and form diploid zygote, which then will develop into embryo. The other sperm fuse with two polar nuclei to form triploid endosperm. So endosperm is an important structure that helps store tissues, nutrients, and for the developing seed. So double fertilization helps to ensure the endosperm develops only in ovules where the egg has been fertilized, and then preventing from nutrients, meaning dia nak elakkan daripada pembaziran nutrients. Provide source of nutrients for developing embryo and germinating seed. Ini contoh ni, ini adalah kacangan corn. And provide growth stimulating hormone for developing embryo. Embryo ni dia akan membentuk biji benih ni, bila dia germinate, biji benih yang germinate tu, itulah embryonya yang akan germinate. After double fertilization, the ovule will develop into seeds and ovary develops into fruit. Yang kita makan, buah apple ni, this is the parts where we eat, which is the ovary. It's the dispersal by wind or animals. Okay, yang ovule, yang ni ovule, seed. Develop into seed. Jadi seed nanti akan bercambah. The integument becomes the seed coat, outer become the testa, inner integument become the tegment. Tegment and testa is lignified, jadi dia keras dan juga kalis air. Strong and durable for the seed coat protection. Contoh kacang hijau itu dicotyledon. Kacang hijau, bahagian struktur yang hijau tu, itu adalah tegment. Outer layer. The testa. Manakala yang bahagian bahagian dalam melaputi seed, melaputi cotyledon ataupun endosperm itu adalah tegment. Okay.